Welcome back. Today we will look at a very interesting aspect of network biology, which is the reconstruction of these networks. How do we build these networks in the first place? Given a network, we have looked at various ways to analyze them and so on. But how do you build this network in the first place? How do you construct? How do you pick the nodes? How do you pick the edges? What kind of meaning do you assign to various edges and so on? So this is what we will look at in today's class. So how do we reconstruct different kinds of biological networks? Reconstruction is the process of integrating different data sources to create a representation of all the chemical events that underlie a biochemical reaction network, right? And we say reaction, but this includes all kinds of interactions, right? So everything is sort of interaction and a reaction, and this is essentially the integration of information from a parts catalog, and reconstruction is the first step towards modeling. So if you want to do all this characteristic path length analysis, degree distribution analysis, or whatever kind of analysis that you want to do on various kinds of biological networks, of course we will see other applications which involve building very complex networks in terms of like a pandemic spread, like what person goes to what place at what time and so on. So all these require a different kind of data sources and things like that. But here we are looking about classic biological networks. So looking at protein-protein functional associations, metabolism, signaling and so on. So let us first start with gene networks, and you see them typically referred to by different names. So gene regulatory networks as they are usually called arises from the fact that the expressions of different genes is controlled by transcription factors. So the amount of a single gene is controlled by the levels of several other genes. So this is the sort of network that exists within a cell, and these transcription factors are proteins, but you can essentially think of them as other genes. So if you project everything to the gene space, so to speak, and the regulatory relationships between genes form really complex networks, and you can elucidate them through experiments such as ChIP-seq and so on. So I will not get into all the biology, but you can obviously read about it if you are interested. And transcription factors are known to bind at very specific sequence motifs. So these kinds of interesting algorithms to identify what are these motifs and things like that, you will be studying it in the other course which is being taught by Professor Manikandan, and there are very interesting algorithms to uncover these kinds of motifs and things like that. We obviously spoke about a different set of motifs in this very course, and network reconstruction involves the identification of regulatory relationships in a given organism. How do you identify which node, which gene activates or represses another gene and so on? Okay. So what does the regulation of gene expression entail? The regulation of gene expression is primarily mediated by transcription factors, which are essentially proteins, which recognize and bind specific nucleotide sequences which affect the transcription of nearby genes. So you may have studied the lac operon and so on, or if you haven't, it is a beautiful example to go and look at as to how when an inhibitor is present, it does not allow the transcription factor to bind and so on and so forth. So when glucose is present, this operon behaves in a different way. When glucose is absent and lactose is present, this operon behaves in a particular way. So these kinds of interesting regulation of gene expression activities can be studied or modeled again using networks. And to date, lots of information has been accumulated in terms of what are the transcription factors and what are their target genes. So this TF-TG interaction is what we look at, and this has been looked at in various prokaryotic systems. So a lot of information has been accumulated on these regulatory interactions between TFs and TGs, the transcription factors and their target genes in various model systems such as E. coli, Bacillus subtilis and so on, and today you have this for a huge number of organisms, and we are also trying to understand the same thing in much higher organisms as well. You have transcription and regulatory networks within a cell which include only the gene regulation mechanisms via transcription. There is only a small fraction of mechanisms by which gene regulation occurs, but again one of the most well studied and still fairly important way by which gene regulation occurs. So what are transcription factors? Crudely, you can think of them as gene products that control whether a gene is turned on or off. So when this particular transcription factor is present, this gene is on. When it is not present, this gene is off. So these kinds of interactions we can capture. And there are regulatory proteins, there are specific regulatory proteins which in turn regulate the transcription of a particular gene either positively or negatively. And there are many databases such as DBD, Jasper, Transfac, Regulon DB and many more databases have since come. This data is a little old, but you see that there are a huge number of transcription factors that have been identified in so many different species. And you typically, when you read papers or when you dig around in literature to identify interesting problems to work on, even for your assignments or projects and so on, you find that there are so many different networks that people talk about. There are gene regulatory networks, gene expression networks, co-expression networks, genetic regulatory networks, TRNs, genetic interaction networks and so on. So all of these capture various meanings, and sometimes we just talk about gene networks, which is like a sort of union of all of these concepts. So you have a huge number of terminologies that are used to capture these in literature. So let's look at an interesting example network to fix certain ideas. So what is it that we are looking at here? We are looking at genes, proteins and metabolites. So these are the molecular constituents of the cell and captured as nodes in your network, and they are organized in 3 levels. You have mRNAs, proteins and metabolites. So think of these as 3 different spaces. You have the gene space at the bottom, you have the protein space that comes right after, and then you have the metabolic space. Well, this is really not how there is no hierarchy or of any sort like this inside the cell, but this is just for our own understanding that you deconvolute all of these to different layers so that you can better understand them. And so what does this show? So the solid arrows indicate interactions, and 3 different mechanisms of gene-gene interactions are shown here. So the regulation of gene 2 by the protein product of gene 1, right. So this is the interaction that is shown. So there is an interaction of this sort, but it actually comes from the other space, so to speak. So gene 3 produces protein 3, gene 4 produces protein 4. These 2 come together to form this complex, which then does something to gene 2. So this basically means that gene 3 and gene 4 interact with gene 2. We will see that in a moment. I will keep these edges away because they will be there in the final slide. And then there is a regulation of gene 4 by metabolite 2, which is actually produced by protein 2. So which means that gene 2 is also in a roundabout fashion connected to gene 4. And now you project all of these. So projections of all these interactions into the gene space is indicated by dashed lines, and this is your gene network. Now you see that gene 2 is very important because it is highly regulated by gene 1, 3 and 4 and things like that. So you have all these kinds of interesting inferences that you can make, which is not very obvious when you look at this more complex picture, right. But the moment you switch to this picture of the gene network, you are able to see a lot more and appreciate what is actually going on in the network. So what are these gene regulatory networks? Any network needs to be defined by its nodes and edges. So in a gene regulatory network, the nodes are genes and the edges represent interactions or modulations. You have even self loops here, so you have n square possible directed interactions here, not just n squared minus n that we typically have, right. So the diagonal is usually left out in the adjacency matrix. If you recall, diagonal is all zeros and you only have off diagonal entries, so it's n squared n into n minus 1 or n squared minus n. But here you actually have n squared possible interactions because you also have self loops. This is because most genes will have a negative effect on their concentration because the degradation rate of their mRNA is proportional to the concentration. So higher the gene, faster the degradation, higher the amount of mRNA present, faster its degradation and so on. And these are the kind of interactions that you see. So gene 1 inhibits gene 2 because that is what protein 1 apparently probably does, right? It's not expressly mentioned here, but we can assume things like that. And then gene 3 activates gene 2, gene 4 activates gene 2, and gene 2 again has a negative effect on gene 4. So this dashed end arrowhead represents a negative effect typically. Okay. So if you see, the edges include automatically or implicitly the effects of both the protein space and the metabolic space. This is very interesting, and as I was saying, you cannot really infer all of them if you are not looking at the bottom part of the screen. If you look at only this part, in fact, even if you looked at the whole part without these dotted lines, all of these interactions don't just directly jump at you, but whereas now you are able to see them much more clearly. So what is the structure of these networks? Especially let's talk about transcriptional regulatory networks. What is their structure? This is a classic review from long ago, where the assembly of regulatory interactions linking transcription factors to the target genes can be viewed as a directed graph, right? So you have regulators and targets which represent nodes, and the regulatory interactions are the edges. And this results in a super multi-layered system which can be examined at 4 levels of detail. What are they? The basic level, where you just look at the network comprising a collection of transcription factors and the downstream genes and the binding sites in the DNA, and these basic modules are then organized into motifs and recurrent interconnection patterns, right? That's what we said a motif is, which appear frequently throughout the network. So you can have like a bifan, a feedback loop, and things like that. And these motifs then cluster into higher order modules, right? And we've already seen we will be seeing applications of disease modules as well later on in the course. And at the top level, you have the entire regulatory network, which consists of interactions between these modules. So you have basic edges, basic nodes and edges at the lowest level. Then you have motifs. Motifs come into modules. Modules interact with them in a complex network. So this is how incrementally you have a very, very complex network. It's not that the network evolves in this fashion, but this is how we can break down a complex network and study it for greater clarity. So here you go. So the basic unit comprises the transcription factor and its target gene. So these are basically just the edges, right? So you have a TF and a TG. So you have a feed forward loop, multiple input, single input, and so on. Various kinds of motifs which are organized into complex modules in this fashion, ultimately into a real complex transcriptional regulatory network. And all of these have been incrementally identified for various organisms. So these are readily identified through experiments, but the moment you start going beyond that, you need to have excellent algorithms also to try and identify this with great care from a background of lots of possibilities. The inference of gene regulatory networks is a very interesting problem in computational biology. So what is the goal? The goal is to elucidate hidden regulatory events. You can think of this as a sort of link prediction that we will cover at some point in the course. And network inference is more than just clustering, right? You just don't cluster genes that co-express together and put them together and so on. But you need to really identify a lot more. For example, you try to build these inferences from different kinds of data, right? So microarray data or RNA-seq data, which is very commonly used, or literature, wherein you just do text mining to find what are the genes that are co-mentioned in various abstracts and so on, and network comparison between two species to identify missing interactions and things like that. And there are various other methods that people have developed based on mutual information, various statistical methodologies, and there are a bunch of algorithms as well for inferring GRNs. So you have, this is typically data driven modeling, and how do you, given this data, how do you actually infer these networks? And this is going to be covered in the last part of this course by Professor Nirav Bhatt. And another very interesting set of interactions arise from synthetic lethal deletions. So these are typically called as genetic interactions, and this interaction is interesting. So, consider two genes ABC and PQR. When you delete ABC alone, nothing happens to the cell, you get a non-lethal phenotype. When you delete PQR alone, nothing happens to the cell, you get a non-lethal phenotype. But when you simultaneously delete both ABC and PQR, you get a lethal phenotype, meaning they were probably compensating for each other or whatever. So, ultimately you cannot rid the cell of both of these two genes simultaneously, and this is quite interesting because it has applications in for targeting cancers. There is this notion of what is known as collateral lethality in cancers, and it's also used for trying to predict combinatorial drug targets against various infectious diseases and so on. And there are various ways to predict these in metabolic networks and things like that. But this is a sort of subtle interaction in that these two genes need not necessarily occur together or bind or produce proteins that bind together and things like that. But when you remove both of these independently, nothing happens, but when you remove them together, the cell dies, right. So, even if they may be in far off pathways, they may be helping to produce the same molecule that is required for cellular survival and things like that. There is a lot of work that we have also done on synthetic lethals in our lab, and if you are interested, you can go and read a little bit more about it.